So in our last screencast, we just kind of did an overview of cells and just kind of what they were, why they're needed, why they're a thing. Today we're going to kind of zoom in on exactly what a cell is, like what are the different parts that make up that cell. So we're going to begin by looking at eukaryotic cells, and maybe hopefully remember that eukaryotic cells, remember, are cells that contain a nucleus. Now, if you look at this diagram here, I don't know about you, I get a little intimidated. There's an awful lot going on in this. And so what we're going to do is break it down step by step to kind of figure out what are all these parts in there. So before we get into that, I want to give an analogy for eukaryotic cells. Think of a eukaryotic cell like a house. Now, houses are compartmentalized. What that means is there's different rooms for different jobs. So I've gone ahead and taken some photos from my home here to kind of illustrate what I mean. So this, for example, is my bedroom. And so this is the place where I sleep and do my morning laps. So that is the room for that job. The function is compartmentalized. The function of sleeping is compartmentalized into that room. Here's a photo from the bathroom. Again, very specific functions for that particular room. A closet is another area, and that is a storage area of the house. The kitchen is for cooking and preparing food. So you kind of get the sense, like there's different rooms for different jobs. Here's the garage where I store my vehicles. So there's different rooms for different jobs. The cell is the same way, only for storing these things, the cell uses, instead of calling them rooms, it calls them organelles, and each organelle has different functions. So in order to break this down, we're going to draw this out. So I don't know if you remember Bob Ross from like the 70s, I think. He like did painting shows. We're going to do a little Bob Ross here, and we're going to draw a picture of a cell together, which I think is going to help leave a fair amount of room. And then I'll kind of be cutting to my buddy, Mr. Sanger, periodically, and then he'll kind of walk through the drawing. So I guess, should we cut over to him right now? Let's cut over. Okay, take it, Mr. Sanger. Thanks, Mr. Sanger. All right, so what we're going to do is we are going to draw a cell. I would encourage you to make this large, like a quarter of a page maybe in your notebook, and then I'm going to draw things and kind of verbally say the name. I would encourage you to write out the name as well. I'm not going to do that just so that the diagram doesn't get too cluttered. So we're going to start by making a big old circle. And this line represents the plasma membrane. The plasma membrane, remember, is what separates the inside of the cell from the outside of the cell and regulates what can enter and exit. And then everything in the interior of this cell is the cytoplasm. We're going to look at the nucleus. So the nucleus's job is that it stores the DNA. And so you may know that the DNA kind of regulates and controls what happens in the cell. And so a lot of times the nucleus is sort of called the control center of the cell. So by storing the DNA, it's kind of storing the molecule that directs much of the cell's activities. The structure of the nucleus, it actually is made up of two membranes, and you can kind of see this here. So one membrane, two membrane, and that double membrane is called the nuclear envelope. And then you'll notice that there's these little holes in there, and those holes are called nuclear pores. So the nucleus consists of a nuclear envelope with little holes called nuclear pores in there. What this means is because there's holes in that membrane, it's kind of like having huge holes in the fence around your military base. If you got big holes in the fence, then things are going to be able to move freely back and forth, and that's kind of what happens in the nucleus. Most molecules are able to move freely back and forth between the nucleus and the rest of the cell. There's really only one molecule that's just too darn large to get through, and that, my friends, is DNA. And that's why the nucleus stores the DNA. It sort of traps the DNA, but it allows lots of other things to come in and out to interact with that DNA. And again, we'll talk more about that later. So let's draw in the nucleus. Here we go. The next structure is the nucleus. And remember, the nucleus has two membranes around it. Those two membranes, it's called a double membrane system. That's called the nuclear envelope. And so to represent that, I'm going to draw kind of like this. like that. And so you can kind of see there's like an inner membrane and an outer membrane here, so it's like a double membrane system. And then those holes are the nuclear pores. On the inside of the nucleus, of course, we have the DNA that's stored there. One final structure I forgot to mention is the nucleus, and that's a site where ribosomes are made. It's often this kind of darker area in the cell. You can see it in a microscope, but it's not a major structure. Okay, next we have ribosomes. Ribosomes are the site of protein production. Maybe you remember learning about protein synthesis in bio last year, transcription and translation, and that took place in a ribosome. That's kind of what's going on here. Again, we'll talk a lot more about that later, but for now, ribosomes produce proteins. The structure of a ribosome is it consists of proteins and a special kind of nucleic acid called rRNA, and that stands for ribosomal RNA. So basically you've got this rRNA, you've got these proteins, you link them together and you make these different subunits that then come together to make these ribosomes. And you'll note that prokaryotic ribosomes are a little bit different than eukaryotic ribosomes. Okay. So next we have the ribosomes. The ribosomes are very tiny cellular structures, so I'm just going to draw them as little dots here. And in reality, they're kind of scattered all over the place, but I'll just cluster them over there for tidiness purposes. Now we have a group of structures that collectively are part of what is called the endomembrane system. It's a system of membrane-bound organelles that kind of interact with each other and work together to kind of do things. So the first one is the endoplasmic reticulum. The endoplasmic reticulum, there's two types, rough and smooth. Rough endoplasmic reticulum, its job is to take those proteins produced by the ribosomes and distribute them to other parts of the cell. So if you think of the ribosome as the Amazon worker, the rough ER is kind of that factory. It's going to take the products of the workers and like ship them off to other parts of the cell. The rough ER is called rough because it has these little ribosomes on it, and so the surface looks bumpy. That's why it's called rough. It's located right next to the nucleus, and the reason for that is the nucleus contains the information that the ribosomes use to make the proteins. So it kind of makes sense that those would be near to each other. The smooth ER, does not have those ribosomes attached to it, hence the word smooth. It's smooth looking. Its job is totally unrelated. It produces lipids, and then it can detoxify poisons. So one example of a poison that might be found in a human body would be alcohol. If someone drinks alcohol, that alcohol is toxic. And the smooth ER in some of the cells, mainly the liver, is going to be responsible for sort of breaking that alcohol down to something that's less harmful. So let's take a moment and draw those out. Next we have the endoplasmic reticulum. The rough endoplasmic reticulum is going to look like this. It's sort of attached to the nucleus, and it's just kind of this sort of weird looking structure there. And the thing that makes it rough, of course, is that it has ribosomes on it. So I'm going to take my red, since my ribosomes are over here in red, I'm going to add some ribosomes to the surface of my rough ER as well. Note my patriotic red, white, and blue theme there. The smooth endoplasmic reticulum looks very similar. I'll put it up here. Kind of that same sort of blobby splat mark kind of look, but there's no ribosomes attached to it, and so as a result, it is smooth. Next we have the vesicles. So vesicles are these tiny, tiny, tiny little membrane enclosed sacs. Their job is to transport materials throughout the cell. So if the rough ER is the Amazon factory and the ribosomes are the Amazon workers, then the vesicles would be the trucks. So they're going to take the protein products and they're going to start to ship them around the cell. And note that they don't just branch off of the endoplasmic reticulum, they can branch off other parts of the cell as well. So their job is to transport materials through the cell. Let's take a moment and draw those in. Next we have the vesicles, and the vesicles are just little transport structures that kind of move things throughout the cell. So here we have 3 vesicles that are sort of branching off the rough endoplasmic reticulum. Next, we have the Golgi apparatus. The Golgi apparatus, it kind of looks like a series of flattened pancakes. Pancakes are good. I have a good pancake recipe. Its job is to receive the proteins and lipids made at the endoplasmic reticulum, and then it modifies them, and then it sends them to the appropriate part of the cell. So here's kind of the picture of the Golgi complex, and you can see our stack of pancakes here, right? And in each of these pancakes, there's different chemical reactions that happen. So, one pancake, you might add a little sugar group to a protein. Another pancake, you might add a sugar to a lipid and make something new. And so you can kind of see, like, um, as we go here, if this is the initial molecule, it's being made in the endoplasmic reticulum, it's altered a little bit, and then when it gets to the Golgi apparatus, the first pancake, it's tinkered a little bit more. Once it gets to the next pancake, it's adjusted a little bit more, and then when it gets to the final pancake, it's changed a little bit more. So the general idea is it's going to modify that product. Next is the Golgi apparatus. So the Golgi apparatus looks like a series of pancakes, really. So I'm going to go pancake 1, pancake 2, pancake 3, pancake 4. And then the vesicle, which is branched from the Golgi apparatus, is going to deliver materials to the Golgi apparatus here, which can then distribute them to other parts of the cell. So I'll make some more vesicles branching off of that Golgi apparatus. The lysosome is the digestive center of the cell. Its job is to break down waste. So take a moment, if we're going to break polymers into monomers, so like chop things up, what kind of reaction do we want to take place? I'll give you a hint, it's either hydrolysis or condensation reactions. And the answer is hydrolysis. So hydrolysis is going to break things down. So the lysosome has these little things called enzymes inside that carry out hydrolysis reactions and break things down. So you can see, here, here's a lysosome. It's got these digestive enzymes, and it's coming over and kind of merging with this food storage structure. And then they're going to merge together and then break up that waste. So let's draw that in. Next we have the lysosome, and the lysosome is sort of hard to represent because it's just a circle. So, here's the lysosome, nothing fancy. There's digestive enzymes in there, but really it's just kind of circular in structure. And the next structure is the vacuole. The vacuole stores materials in the cell. This could be food, it could be pigments from flower cells, it could be all sorts of stuff. Animal cells have lots of small vacuoles, while a plant cell has sort of a single really, really large vacuole that takes up much of the body. So let's draw that one. The vacuole is similar to the lysosome in that it's just kind of a circular structure. So you'd have to label, lysosome and vacuole, and again, they both look very similar to one another. And then just to kind of recap the endomembrane system, note that all of these structures interact. Okay, so in this diagram, we see ribosomes on the rough endoplasmic reticulum are making a protein. The endoplasmic reticulum uses a vesicle to ship it to the Golgi apparatus. The Golgi apparatus is going to modify that protein in some way and then send it to the appropriate part of the cell. So maybe the protein product is a hydrolytic enzyme, a protein that chops up things. Well, then it's going to get shipped to the lysosome to do its job. Or maybe that protein is something we want to store and hang on to. Well, then it's going to get shipped to, whoops, that doesn't say vacuole. It would be shipped to a vacuole for storage. Or maybe the cell needs to release that protein product and get rid of it. So then it'd be shipped to the cell membrane. So this endomembrane system is sort of responsible for shipping and distributing all sorts of materials throughout the cell. Here's another example of that. Food could be taken into a cell across the membrane, and then it's going to enter a lysosome. And yeah, there's some intermediate steps there, but here's the lysosome. The lysosome is going to break the food into monomers, and then the monomers might be sent to the ER so that ribosomes can use them to make proteins again.